And it's my pleasure to introduce our next speaker. He is a professor of physics at the Norwegian University of Life Sciences and University of Oslo. Please welcome Dr. Gaute Einevoll.
Thank you. It's great being here, and I'm a big fan of the Allen Institute. And this is not our institute back in Norway. It's actually the farm where my father came from. And my grandfather, actually in the early 30s, he had to come to Seattle to save his farm because he had guaranteed for some other farmer who couldn't pay the loan to the bank. So Seattle and the area here has a special sort of meaning for our family. No, he went. So this, he had sort of like 5 children before he went to the U.S., and then he was there 4 years making money to pay the bank, and he came back and had 4 more children. So my quite impressive grandmother also I had then, sort of. So I wanted to start with this. This is Abel. He's the most famous Norwegian mathematician. He died in 1829, as you can see from the stamp here. And I read his biography, and he was so upset that he died. He died from tuberculosis just before he got this sort of job as a professor in Berlin, which he had dreamed about. And he was so upset about the doctors. How can they fix this silly tuberculosis thing, right? And now, in our part of the world, we don't die of tuberculosis anymore. And hopefully also the work like people in talking before lunch, that with new antibiotics, we'll keep it that way. But if we go to the doctor today and come back with a problem with the brain, like dementia, or even like some psychiatric diseases, schizophrenia, bipolar disorder, there's very little the doctors can do, or that the medical science has to offer. So, and this is also reflected in this opinion piece about one of the previous directors of the National Institute of Mental Health, complaining about this. All this resource we put in, why aren't we doing better? Why aren't we solving this very, very difficult problem? So I think if you ask neuroscientists about this, they point to all these different scales. It's so hard to connect all these scales. And this is true. It is hard to connect these different scales, all the way from the molecules, all the way up to cells and to the body, even though I think before lunch I was quite impressed by some of the work presented about how modeling has sort of linked all the way up to cells, or at least bacteria, using modeling. But still, this is sort of has been the excuse. And then you can ask, are there other problems, or other issues, other problems we have to solve which involve scales? And there are, and I think the most maybe impressive one is weather prediction. Actually, another Norwegian hero, is now on the tail fin of a Norwegian plane. He suggested in 1900 that weather prediction is a mathematical problem. That you don't need to use all this weather science or statistics or just observations. You can actually compute the weather in principle. Because this was not possible at the time. This is also illustrating that in terms of bridging scales, this is just sometimes similar to what you have to do in the brain. Have to link all these sort of phenomena, weather phenomena, all the way from the 100-meter scale up to the whole globe eventually, if you want to have climate models and so on. And this is now extremely successful. I think the first weather forecast from mathematical models came in 1954. Of course, the first time you could actually do it in real time, so that it could be at least sort of before the weather occurred. So it was useful. It was in the 70s, 80s. But now all weather is computed all the weather predictions are computed, right? And this is the prediction of Seattle, which I printed out in Norway just before I came here. So it seems like the sunny outside. It's winter. Exactly. So, and also this is sort of a steadily improved quality. It predicts one day better into the future per decade. So meaning that now we predict, the weather is predicted with equal accuracy 6 days in advance as 5 days in advance 10 years ago. And this is just a fantastic combination of models and observations and statistical methods, and it's a very, very impressive endeavor, I think. And also smartphone is sort of a little bit similar to how, this is of course different, it's man-made, but also there we have found ways to sort of link all these different scales so that it can actually program the computer and actually do things at least on the transistor level, just with my finger now. I think you also can do that, right, when you push an app. So these are sort of very impressive multi-scale endeavors, and the key here has sort of been the use of mathematics. That's the only the only language which is precise enough, really, to really precisely link these scales. And because I was thinking that I was at this workshop in Copenhagen with Adrienne also a few weeks ago, and then there was this discussion about principles of the brain. What is sort of like, how can we sort of, what is thinking about how to get towards the understanding of how the brain works. And then I sort of realized that there are many suggestions for how, like, principles for how the brain works. And then you can ask, why aren't we making any progress? I mean, some of these suggestions are really old. Ideas are sort of reused and sort of brought up back again. And it turns out, I mean, and the main thing is that it's very difficult to falsify the theories. You can't really make progress unless you can falsify theories. So in order to do that, we need mathematics, I think. And this is sort of like, I would say, like the starting point, just called from this Manifesto of Brain Science by this Breitenberg. We are convinced that ultimately a satisfactory explanation of thought and behavior will be given in language akin to that of physics, that is, in mathematical terms. So, and we actually wrote the paper. I'm involved in the Human Brain Project, and we wrote a paper that came out a few weeks ago in Neuron, which is like a high-profile journal in neuroscience, which was about the scientific case for brain simulations, really about why we need to do mathematical modeling. And I was thinking, this is sort of a strange title. I mean, could you think of it, the scientific case for cell simulations? Could that sort of get into a high-profile journal? I suggest not, because even that we just still discuss this in neuroscience, I think tells us that we are sort of a little bit behind other parts of like cellular science. And one of the main points of this was that there's all this discussion about the Human Brain Project, saying that we don't know enough about the brain, so to start modeling it. And I think one misconception there is sort of the idea that you need simulations. It's not like a one-off thing that you test one model, and if it doesn't work, we all go home and are depressed. It's actually, brain simulations are for testing different theories. And we made this comparison to this Newton's idea about the movement of planets, which was sort of obviously a success. He put up these formulas for the forces and between masses and planets and so on. But in order to test his theory, then he needed to develop actually a new kind of mathematics just to test it. It's very difficult to go, or at least you have to study. I mean, you have to study to be able to go from these equations to predict planetary orbits. And so what we do with brain simulation is a little bit analogous to sort of inventing calculus. We need sort of this. You cannot just do like a paper and pen calculations to sort of test your hypothesis like Newton could for this particular case. And that was sort of a starting point or that was how we could sort of compute all these different things, planetary orbits and ebb and flow of the water, of the oceans and so on. And how this theory was confirmed. So how do we do this in neuroscience? For one thing, if you want to focus on the cortex, which is sort of like a little bit like an element, a pixel of sort of what Peter talked about, a piece of cortex with, say, like 100,000 neurons, what is called a cortical column. We have all these different ways to measure neural activity. So what you do with all this data, you can measure spikes, the electrical measurements, and different kinds of electrical measurements, and you can measure actually magnetic field out here, and there's all these kind of optical measurements. And the typical way to use this data is just to do statistical analysis, try to correlate this data with typically what the animal is doing or what kind of stimulus it sort of is presented to it. And one thing that we have to improve there is sort of to link, if you start with a model, if your model involves neurons like this one, from this and you have neural activity, from that you can compute all these measures. The same model can compute all these different measures. And that means that if you measure all these things simultaneously, we can use all of the data to falsify or validate the same neural model. And this sort of measurement physics has sort of been, not really been, I'm sometimes same thing, we have so few computational neuroscientists that we don't really have time to do everything that is needed. Sometimes I say it's like 10 people trying to settle Australia. It's just too few people. And this thing of measurement physics, linking your predictions from your network, your model, linking your model on network predictions to things you can measure is actually a separate kind of physics which has not been developed, and that's something our group and also the group here at Allen Institute with Christoph Koch and Anton and others have worked on. So this is sort of like the perspective. If you want to understand like a cortical column, say like a piece of barrel, in this case a piece of the cortex that has to do with whiskers, with maybe 100,000 neurons. We don't really know all about, we know we know the neurons and we know that they are connected. We can make like a skeleton model, but we don't know all the parameters. But what we would like to do, nevertheless, is sort of to work towards getting these models at the different levels of resolution, all the from biophysically detailed models and like point neurons and firing rate models, sort of like what Pete talked about. But we also make the connections to these different measurement modalities, because we don't know really how these things is wired up. But we do know if we make an assumption, if you have a model, we have the principles for how essentially we know how to model neurons based on the cable equations, and the principles for modeling both like the neurons and networks is well established. This was an excellent book by Christoph, by the way. It's like a classic in the field, showing how this can be done. But then there's like this second aspect. How do you link these neurons to the things you can measure? And that's a separate thing, which is also now, I would say, developed. So we don't know really how the circuit is wired up. We are able to test the candidate theory, compute the consequences, and then compare with experiments, and thus make progress. For this, we also need tools to do this thing, and we have developed this LFPy. Bionet is developed here, NetPine somewhere else. So that's also something you need to do so that not everybody has to reinvent these computational tools. So, and of course, then we do standard physics. We formulate a hypothesis. We compare, compute the consequences, and then and compute all these measurements, and then do the measurements, and then try to, going from these experimental recordings to go back and say, to identify candidate models which are most compatible with experiments. And this link from neural activity to what you can measure is not so obvious. For example, this is a single neuron that receives an oscillatory current up at this apical dendrite. And you see you get this nice dipolar pattern. And what you measure with the EEG electrode, for example, is like the sums of millions or hundreds of thousands of these neurons, and which each contribute to the extracellular potential. If you do the same thing 100 times...
Driving the soma at 1 hertz and then 100 hertz?
Actually, this is driving it at 1 hertz, but this is it's driving the synapse. But you still get the same kind of pattern around the soma. But if you do it at 100 hertz, 100 times faster, the same current, you don't get the same thing just 100 times faster. You get a very different pattern. And this has a much smaller dipolar contribution, meaning that you will get a much smaller contribution to both the local field potential and the EEG. So this just means that there's all these biases in the having to do with the measurements, which you have to do properly in order to interpret the experiments. And so we have developed these tools. If you want to sort of go all the way up to the human, the whole brain system, we can sort of, if you look at this, for example, a population of neurons which is active, can be measured both locally, here with the LFP and spikes, but you can also measure it with this ECOG, electrodes placed on the cortical surface, or EEG, measured on the cortical, or the brain surface, or even MEG. And this is sort of just an illustration of some of these, this forward modeling. So here there's one population, in this case 10,000 neurons, which gets synaptic input. And what is shown here is sort of what we will measure from this electrode that is placed in the middle of the population, that's shown here. And this is what you would measure from at the top of the head with the EEG and and with the MEG here. So that just illustrates, of course, this is just to illustrate the forward modeling. But the thing is, with these tools, you can sort of put your hypothesis, hook up the network you want, and then sort of compute the consequences and compare with experiments. So it's like a starting point for doing experiments. This is an example of where we have took a piece of cortex, 80,000 neurons. We just took a model called the Potel-Stiesmann model. And in this case, it's like this, it's 80,000 neurons. This shows this is like receiving input at, receiving input at 900 here. It gets this input from the sensory system, so you get this strong, like a spike of activity across the cortex. And that you can now compute not only the spikes, but also the LFP and the EEG or whatever. So then, of course, then you can try with different candidate models and sort of compute all these things, and then from that go backwards and try to do the measurements and say what of these candidate models are closest to experiments, and thus make progress. So just briefly mention, we have started now to do using this, because we need a starting point model. And the state-of-the-art starting point model, I would say, at least, is the mouse V1 model, which has been developed here at the Allen Institute, 250,000 neurons, 21 neuron types. So now, of course, this is, I would say, like a starting point model. We now try making predictions, sort of like using that model to make also prediction about local field potentials. And and here we sort of, this is done a few weeks ago, where we actually looked at this, computed the local field potential, and this model was then tuned just out and was just tuned to predict the spikes correctly, following some different types of visual stimuli. And here we computed this local field potential, which is a low frequency part, reflects very different aspects of the neural activity. And the short take-home message is that this initial model, the LFP you measure and the LFP you predict is quite different. So there's obviously more to do, but it's sometimes a good news also. It means that this signal can also be used to falsify models and find out what is the correct one. I want to end up with just this final graph here, that there's one revolution which has happened, obviously, is the genetic revolution, that you can now measure the genes of extremely cheaply for big cohorts of people. And so we have started also to use like the GWAS information from GWAS studies of schizophrenic patients to try to see how these the genetic variations actually affect the neuronal behavior, and actually all the way up to networks and actually up to even EEG signals, which is one of the few things you can measure actually on humans. So that's what I plan to say.
We have time for a question or two. A microphone is coming to you shortly. Excellent.
Can you tell me basically what a seizure is?
Yeah, a seizure is sort of like in the brain you have this competition between cells, excitatory cells, and inhibitory cells. And excitatory cells try to get the neurons they communicate with, other neurons, to start firing action potentials. The inhibitory is like the brake, so it's like a gas and brake. And if the gas pedal wins, then you can get this like runaway electrical excitation, which is sort of then a seizure.
In the middle there, right behind. There you go.
Hello. I like that V1 model. If I wanted to run that, what kind of compute resources do I need? Is that on-prem, or is that in the cloud? Is it a laptop?
So there's you can ask Anton or Stefan about this. So I think there's two versions of the V1 model. One which you can run on I would say like a normal laptop with if you have like some memory, and the other ones you need sort of like many clusters, like a supercomputer. Oh, how super. Yeah, hundreds of CPUs.
Excellent. We're going to have to cut it off there, but thank you so very much.